This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 3. Hey everybody, it's Vincent Racaniello, and I'm with a smiling Dick Despommier.
Hey Vince.
How you doing today?
I'm doing very well.
I hear you had a busy day.
I did. But busy is good.
You taught a lot, right?
I did.
Gosh, you look a little exhausted.
I feel...
Why is your face red?
Because I just ran over to your studio.
I see. Parasitology. What did you talk about in parasitology today?
Today in parasitic diseases, to our second-year medical students, we presented malaria.
Which will be a future topic on this podcast.
You bet. And a frequent topic, I might add also.
Excellent. And what do we got for today?
Today, I thought we would discuss the difference between intracellular and extracellular parasites, with an example starting with an intracellular parasite of an unexpected size and character.
Intracellular versus extracellular
That's
parasites.
right.
The extracellular, did you call those ectoparasites last time, or is that not correct?
No, an ectoparasite is something that lives on you, like a head louse or a body louse, and they're considered ecto because they're on the outside.
God, I know the head louse business. The kids always pick them up in school.
Absolutely.
So those are ectoparasites.
They are.
All right, so what's the one that lives outside of you that's not an ectoparasite?
There are temporary parasites.
They do attach to you?
Yep. In fact, body lice are like that. Body lice actually live in your clothing. And the only time they take a blood meal is when they jump onto you, and then they come back off again.
In that book that you loaned me,
yes,
Rats, Lice, and History,
yep,
tells the story of Thomas Becket, who, as you know, was murdered in Canterbury Cathedral.
This is true.
And as he lay in state with all the lice came out of his body, he had apparently lots of layers of clothes on. It was cold, and it was full of lice. Cause I think they didn't bathe very much back then.
No, they didn't.
And as his body cooled, the lice boiled out of his clothing. What a sight. That
boiled.
It's amazing, isn't it?
Yes, it certainly is.
Boiled. Well, so you go ahead. I'm interrupting.
No, you're not at all. Not at all.
You want to distinguish between parasites which live in cells and outside of cells.
That's right.
So what are you going to start with first?
Well, I'll just give some examples of each, and then I'll jump to the organism that I spent 27 years of my research life looking into.
Jump? Is that a...
Jump. I jump. I'll
parasites
jump.
do that. All right, go right ahead, and then I'll ask you questions.
So, as I recall, the last time we talked, we were illuminating various groups of organisms, and the groups that I wanted people to start thinking about in terms of eukaryotic parasites, at least, were the ones that were short-lived and ones that were long-lived. Because the long-lived parasites have lessons to teach us that we can apply to problems that have nothing to do with the parasitism itself. So I call this a field that has yet to be developed. I call it parapharmacology.
Did we talk about that last time?
Yeah, we mentioned a little bit about it. And so it turns out that there are two other kinds of parasites to discuss as well, and that is intracellular versus extracellular. So,
all right.
Eukaryotes have lifestyles that occupy both of those extraordinary sites. The extracellular, of course, could be any site in the body. A lot of them are found in the intestinal tract, for instance.
Sorry, you're talking about intracellular?
No, the extracellular.
In the lumen of the intestinal tract.
Or attached to it with
that's
their
considered
device.
extracellular,
It
of
is, of course it is.
Okay.
And then we have some that, of course, live within cells. And we distinguished between multicellular organisms last time and single-cell organisms as well. So a lot of the single-cell organisms, not all of them, but a lot of them, turn out to be obligate intracellular parasites. So they share a lot of common features with the viruses that we talk about on TWiP.
Is that because they're single-celled, they can't provide everything they need?
Not necessarily. I mean, there's some that live outside the cells, like Giardia, for instance, and Entamoeba histolytica, for instance, and a lot of
they can multiply outside of the cell.
Absolutely. absolutely. Yeah. But there are others that are obligate intracellular. For instance, all the Leishmania, for instance. And interestingly enough, American trypanosomes, namely Trypanosoma cruzi, turns out to be an obligate intracellular parasite. But its mate that it split off with 65 million years ago, or perhaps even longer ago than that, when Africa and South America separated. African trypanosomes are extracellular. They live in the bloodstream or in the cerebrospinal fluid, but they don't live inside cells. So here are two trypanosomes, probably from a common ecological or evolutionary, ancestor, which occupied this large single continent. And then when the continent started to break up, the parasites differentiated and developed different lifestyles, but they're both vector-borne.
So when these were first discovered, why weren't they separately classified? Because they had such different requirements.
Because when you look at the stages that circulate in the blood in order to transmit themselves to another vector so they can transmit to another human, those stages look basically the same. So the trypanosome stage of T. cruzi and the trypanosome stage, and there's only one stage of the trypanosomes that are found in Africa, although there are other terms for them if you get into the vectors, they looked very similar. So there's no reason to say that they're going to be different, but as it turns out, their lifestyles are totally different.
So two different trypanosomes, one intracellular.
Correct.
One extracellular.
Exactly. And then we have lots of you'd call them flagellated protozoans because at some stage they do have a flagellum. But at the stage at which they infect us into our cells, they lose that flagellum and they become an amastogote, or a parasite that does not have a flagellum. And that's the Leishmania. The Leishmania do
These are extracellular guys.
Those are intracellular.
Those are intracellular?
Intracellular.
All the time?
The Leishmania, yes.
Why do they have a flagellum then if they're intracellular?
Because in the sandfly host, they're not all intracellular.
I see.
So they have two different phases to their life, but when they're in us, they're intracellular.
All right, so Dick, I'm getting confused. It's too much. We got to classify this for the simple-minded virologist.
We had a similar discussion this morning in the medical school class about malaria and all the nuance of its life cycle, and compared that to viral life cycles. And the difference is that people that come into medical school come in with a background in cell biology, so they understand the mechanisms that viruses use in order to replicate. But the mechanisms that these protozoans use to replicate are totally different. And,
well, they seem to be more diverse as well.
And they're much more diverse.
Do we need to know whether some are intracellular and others are extracellular? Is that important?
I think when you start to consider their disease production, the answer is absolutely.
So for today, we're just going to say there are some that are intracellular, some that are extracellular.
That's right.
And leave it at that.
And it cuts across both protozoans and helminths.
They can be in either category.
That's correct.
All right.
So what I'm going to tell you about today is the intracellular parasite that I worked on for 27 years, which is also a worm.
So I was just going to say, can worms get into cells? Because they're big, but a 6-foot worm is not getting into a cell, obviously. So these are pretty small worms,
They're pretty small, but nonetheless, they're still larger in length than the cell type that they infect. So they are intra-multicellular.
They go into more than one cell.
In one phase of the life cycle of this parasite that I'm about to describe, the answer is yes. But there are some pretty big cells in your body, Vince.
Yeah, my brain cells are really big.
You're not kidding. Vince is a very smart guy, by the way.
I'm not.
Sure he is.
I'm just joking. But it's true, actually. Neurons are big and long.
Huge. Now, can you think of another big cell in your body? I'll give you a hint.
Muscle cells?
Exactly.
They're quite long, right?
Striated skeletal muscle cells are big, very large. And they're multinucleated. So if I were a parasite and I wanted a nice, comfortable life...
You are a parasite.
According to our dean. No, let's not go there.
You're an extracellular one.
I'm an extroverted parasite. So I studied a parasite called Trichinella spiralis.
Trichinella spiralis.
Right. And the name actually is a Latin term, which means... Trichinella means little hair.
So this was given to it when it was first observed in the microscope?
That's correct.
When would that be?
I will talk to you about the history of this in a moment. And the spiralis, of course, refers to the fact that when it's inside of its host cell, it curls up into a spiral.
Would that be the picture on our badge?
It would be. It would be, in fact.
Okay.
That is.
We'll get to that, right?
We will get to that, right? But so for all of these diseases, and I'm sure this is also true for the viral diseases as well, and bacterial and fungal, they all have a rich history of discovery.
Mm-hmm.
And in order to fully appreciate how we come to know all of this, I would like to tell you a little bit about the history of discovery of Trichinella.
I'd love to hear it.
Good.
And I'm sorry if I interrupt.
Please feel free to do so. No,
right.
No. I want you to. In fact, you are the person that represents the audience that asks the questions that they're dying to know the answers to. So let's begin with the city of London. Okay, the city of London is the scene that I would like to set for you, and it's the year is 1835. And London in 1835 is one of the most exciting places in the world to be, and it's also a cesspool. It is filthy. It is overrun with rodents. It has received from the East India Trading Company bilge water from the Bay of Bengal, which is laden with an ecosystem that will erupt into a cholera epidemic of major proportions. And it's because there's sewage in the Thames, and there's tons of dirty water being drunk, and the Broad Street Pump is, of course, everybody knows that famous story now of Jon Snow and the Broad Street Pump. And in 1835, London suffers.
Is this before we understand about infectious diseases,
it is,
right?
It's at the beginning of the discovery of infectious diseases.
So Lister hasn't done his thing.
Nope.
Pasteur has... Leeuwenhoek has already found the animalcules in a microscope.
Correct. That is correct.
Pasteur has been working, right?
Yeah.
But the germ theory of disease, which was
Koch,
not yet.
It's brewing, but it will depend upon the isolation of cholera and the identification of it in order to move forward. But a lot of other infectious diseases have been thought about, have been speculated about, but certainly the worms have been known about for a longer time because they're bigger, they're easier to see. So tapeworms have been known about for a long time.
Mm-hmm.
There's some wonderful historical stories about the discovery of how tapeworms are transmitted and what they do.
And we'll hear those in the future.
We will.
Can't wait.
I'm dying to tell you. Vince, you'll just love them. They're remarkable stories in their own right. But the discovery of Trichinella has as its background the British Museum and the director of the British Museum, who was a man by the name of Richard Owen, and another man who became famous. His name was Paget.
Okay,
James Paget was his full name. And in 1835, I will recommend, by the way, I have a pick of the week.
Good boy.
Which I can go to right now. It's a movie. Okay. And it's a movie about acquiring bodies for anatomy lessons in 1835.
So medical students, right?
Yes, it's a movie about that, and when I recall the title of it, I'll tell it to you.
Okay, no problem. You have time.
We have plenty of time. So maybe I'll say I know it, but I won't tell it to you right now. So the point is this. The director of the British Museum had access to a microscope. And it's funny you should have mentioned Leeuwenhoek and his microscope, because that was back in the 1600s. This is now in the 1800s. And now microscopy has advanced quite a bit since the discovery of how to handle lenses. And in fact, there were only, as far as we know, only 2 microscopes in London. And one of them was at the British Museum. The other microscope was owned by a man by the name of Brown. Now,
Harold Brown, your boss.
He wasn't that old. But you'll know him by his motion. That's a hint. Can you think of somebody who...
Yeah, Brownian motion.
That's the guy.
He discovered Brownian motion.
He did. He was a physicist.
By looking in a microscope at things moving.
That's correct.
I see.
That is
Particles of dust being pushed around by...
Well, that was Tyndall.
Okay.
Tyndall looked through the window and saw the particles of dust as soon as the light hit them, and you could see them move around. And Brown, with his microscope, saw the same thing under the microscope that Tyndall saw in the middle of the air.
What did he look at under the scope?
Just water with particulates in it.
Particulates were moving around.
Their charges repelled each other, so you get this kind of a random, chaotic movement.
It's not the atoms knocking into them? It's the charge.
It's the charge.
And that's Brownian motion.
It's called Brownian motion.
Okay.
So he studied that under the microscope. Can you imagine devoting your whole life looking through a microscope, watching little particles jump back and forth, and then writing these wonderful treatises on them?
Depends what else you do in your life, I suppose.
This is all
If you can go home and have a good meal, it's not bad.
Well, it depends on what you eat.
That's right.
So Paget, being a first-year medical student, was obliged to attend anatomy lessons. And anatomy lessons were conducted over autopsies. And in those days, autopsies occurred on a regular basis. But the movie that I'm thinking about, the name of the movie is The Devil and the Doctors.
The Devil in the Doctors?
The Devil and the Doctors.
Okay.
The Devil and the Doctors. The Devil and the Doctors. And it was about acquiring bodies for anatomy lessons. From grave robbers, of course. Well, in this case, Paget shows up for his anatomy lesson, and it's a 51-year-old bricklayer who had died of tuberculosis. And we know all this because Paget kept accurate records, and he sent letters to his brother, which we're fortunate to have access to, because it explained the difference between how he discovered this worm and how Richard Owen claims to have discovered this worm. Okay, so here we have Paget attending this autopsy, and the surgeons, of course, without gloves, of course, are digging into the cavity.
They're in their suits and ties, right? No lab coats.
That's right. And this guy has just died from cavitary tuberculosis, and they're opening up the chest with scalpels, and they're pulling apart the rib cage and showing them the lungs, and they've got their bare hands, and I think the mortality rate among surgeons must have been tremendous. And you nick yourself, and of course you're susceptible to all these infectious diseases, and it's just an awful thing. And then they break for lunch, and they don't even wash their hands because Semmelweis's papers haven't come out. It's just an awful mess, right? So Paget's way in the back of the room, and all he can hear the whole time, he says, is them cursing at the dulling of their scalpels. Now, the surgeons had to sharpen their own scalpels. Okay, and there weren't these disposable jobs that we have today. These were beautifully done surgical instruments, right? And they were dulling. Every time they would cut through a layer of muscle, these things would dull up. So what the hell's going on? And then finally they opened the chest cavity, and the guy took his hand, according to Paget, and he ran it along the surface of the diaphragm. And he says, I knew it. It's another case of sandy diaphragm.
Sandy diaphragm.
Sandy diaphragm.
That's what's dulling the blade, eh?
Right. Something about the grittiness of the muscle tissue is dulling the blade. So Paget, being the inquisitive youth that he is, raises his hand, and reluctantly one of the surgeons acknowledges him, and he says, "Yes, Paget, what is it this time?" I could just hear them saying it. "What is it, old boy? Make it quick, we're late for lunch." And Paget says, but sir, could you please explain what sandy diaphragm is caused by?" The surgeons looked around and says, "Are there any other questions? And if not, then have a good lunch, old boys, and see you back here at 2:00.
Nice.
Right. So everybody left the room, except Paget. So what did he do?
He went down to the corpse and put his hand along the diaphragm.
He was smarter than that.
He took a sample and looked at it under his microscope.
He didn't have a microscope.
He stole one from Brown. No, he went to Brown.
He did.
Okay.
That's exactly what he did. Knowing that Brown had a microscope, this guy was widely read. By the way, Paget's disease is named after him. He became a very famous pathologist afterwards.
What is Paget's disease?
It's a degenerative disease of the spinal column.
Okay, it's not infectious.
No.
Okay.
No. This is nothing infectious at all.
But
James Paget later on became Sir James Paget.
Wow.
He was knighted.
So he took a sample from
from the diaphragm tissue and put it into a little vial and rushed off to the British Museum. And in the meantime, someone from Richard Owen's bailiwick had heard about the rumor that there was this corpse with sandy diaphragm, and someone had asked this question. And Owen says, "Get me a piece also." and bring it back here. So that's what happened. And so Paget had to go all the way to the British Museum for this, right? So Owen had his emissaries show up and grab a piece and bring it back. And Owen actually had access to a microscope as well.
Mm-hmm.
So James Paget rushes in, knocks on the door of Brown's office and says, Sir, may I borrow your microscope? And he says, for what purpose? And he said, now, I didn't tell the whole story correct. Okay, so Paget actually, cleverly, with this piece of tissue, had 2 glass slides at his disposal and actually put the piece of muscle tissue between the 2 glass slides and squeezed.
Mm-hmm.
All right, and with a hand lens, he was able to observe parts of the structure.
Mm-hmm.
And what he saw actually amazed him. He saw tiny little coiled up objects that moved. Isn't that
with his naked eye?
With his no, the aid of a hand lens.
Hand lens, okay.
Now, that's not a microscope, but at least it gives you 5 or 6x magnification. Now, the other thing he noticed was that a lot of them were opaque and looked to be white, basically, and so that may be calcified. Who knows? And that's we know now, but he didn't know that they were calcified, but indeed they were.
That's why they were rough.
Yes,
and
that's
correct.
Why they
dulled
the knives, and they would,
and and his body was infested with them.
He died with them, not because of.
Yes, but he had lots of them.
He had lots of them, is right.
Wow, was that common?
Well, in those days, it was, in fact, as it turns out, because sandy diaphragm was very common. Interesting, right? So, young Paget says to the owner of the microscope, he said, Sir, I would like to observe this specimen under your microscope. And he says, and what do you think you'll see? And he says, well, I think that I shall see worms. Have you ever seen worms under your microscope? And Brown was remarked to say, no, thank heavens, which is a great response, right? So, we almost have a conversation between these two. This is a senior member and this is a first-year medical student, and how dare he do such a thing? But Brown was absolutely entranced with this, and so he said, please feel free to look at it underneath my microscope. So he did, and lo and behold, he saw what we use now as our icon for This Week in Parasitism. He saw that object, not quite as beautifully portrayed as that, but it's still quite beautiful.
Amazing.
Yeah, it's amazing. So not only did he see a worm, he saw the worm actually move. So he rushes home, and he immediately starts to write about this to his brother. I saw the most amazing thing today. I got a piece of tissue from a dead Italian bricklayer, and he had died from tuberculosis, but the consumption, they used that word then. But I discovered in the sandy diaphragm tissue these minute worms that may or may not have caused disease in this person, right? But in the meantime, of course, Richard Owen, looking through his microscope, which was a better microscope, was able to see more detail and made drawings. And then the story gets muddy. Okay, the story from now on gets muddy because Richard Owen presents his data at the Royal Society. And James Paget was able to present his data to a student science meeting that was held as part of his medical school education. And neither one of them knew about the other. So it's not certain that Owen stole this from Paget or Paget stole it from Owen. We're not sure who made the original discovery, but if certainly Richard Owen being a more notable person. And why do you think he was notable? Why was Richard Owen such a notable person? And I'll tell you. I'm glad you asked.
I have no idea.
I'm so happy you asked. So Richard Owen is a famous personage in history of the history of evolution. Let me just give you some background. Richard Owen coined the phrase dinosaur to indicate any ancient animal of a certain horrific and the word dinosaur in Latin means horrible lizard.
Yes, right.
And of course, we now know that they were not lizards. They weren't even they weren't reptiles.
They're birds, right?
They were some kind of a warm-blooded thing.
They probably had worms also.
That would absolutely. And the other thing he did, of course, was to in public directly oppose Darwinian evolutionary hypothesis. And confronted Darwin repeatedly in public debates and used special creation as I can believe all of what you're telling me with regards to everything except humans. And Darwin said, why should we be different? And as the proof, of course, one day, lo and behold, and I think I've described this on an earlier show in TWiP, the brain of a gorilla shows up in the possession of Richard Owen. And so Richard Owen immediately gets out a preserved human brain and starts to compare, believing full well that the gorilla brain has to be different. And he can't see a single difference.
It's not much.
It isn't different. In fact, it's almost identical. So what do you think Richard Owen does? He reaches into his drawer and he pulls out a scalpel. And he starts to alter the gorilla brain to make it different. And he removes a little piece here and a little piece there, and pretty soon, now when you look at them, they are different. Then he decides to hold a public lecture and disprove Darwinian theory once and for all, and of course he's helped out by a bishop. And here he holds these two brains, and you see, I told you, humans could not have possibly come from apes. Here is the first ape brain, and look at how different it is. This is a gorilla, newly discovered in Africa, and here's a human brain. It's totally different. So, Vince, who was called Darwin's bulldog? It was Aldous Huxley. So Aldous Huxley, who was absolutely incensed over this irate know-it-all that was wrong, got another gorilla brain, a second gorilla brain, and compared it to the first gorilla brain and was able to show conclusively that the first gorilla brain was surgically altered. And the only person who could have possibly... Now, remember that series on television, Columbo? Now, you know, I just want to ask you one more thing here. He says, you got this here brain, and it looks okay. He says, but it doesn't look quite like this brain now, does it?
Pretty good, Dick. Yeah.
I even have a ratty old raincoat that looks like his. So the point of this whole thing is, of course, that Huxley now has another public debate with Richard Owen and confronts him with his surgeries.
Right.
And says, you're a fraud. You've defrauded the public. You're absolutely a fake. And you're a thief of knowledge.
Terrible. Really terrible.
Really terrible.
But science is self-correcting.
Correct. And this is how.
That's it. Someone is always going to find the truth.
So
right?
Poor old Richard Owen he lost his position, of course, at the British Museum. He became a broken man and was relegated to the distant history books of biology in terms of discoveries. However, he was the one credited with discovering Trichinella spiralis. So that's my intracellular parasite. That's the kind of story that I guess has kept me interested in the subject all these years. So what is Trichinella anyway, and what does it do to you? And there's there isn't time in this segment to go through all the details of this infection.
Oh, we can cover it in multiple segments.
We can do that.
No problem. Go as far as you'd like.
Okay, so the infection was then discovered in lots of other animals, in fact. When they started to look at surveys of animal tissues, indeed, almost all the carnivores had it. All the dogs of London had it. All the cats of London had it. Pigs even had it.
Cows?
Cows are herbivores. So cows...
All the carnivores had it.
Right. Meat eaters. All the meat eaters. Not that they couldn't catch it. It's just that they didn't routinely eat meat.
Got it. So birds don't have it, right?
Birds don't have, although there is a special species now that birds do catch. So I'll talk about that later, because you can have carnivorous birds as well, birds of prey. So in those days, there was only one species known. That was Trichinella spiralis, or at least they thought they only had one species here. And that infection was then worked on by a number of very famous people. And Rudolph Virkov was one of those people in Germany that actually worked on the life cycle and actually determined the life cycle, and then would tour the countryside of Germany warning people not to eat uncooked ham. That was a famous dish.
How did we know that it came from uncooked ham all of a sudden?
Because he was able to trace this infection in people. By that time, Zenker, who was a physician, had discovered the fact that this can cause illness and even death. So it can be a lethal infection in people. And he traced it back to these meals that people had consumed of pork that was lightly smoked and salted, but not enough to kill off the worms inside.
But do pigs eat meat?
They do, in fact. They will eat scraps of other pigs if you feed them to them. They will eat rats that they catch in the barnyard, for instance. So they're omnivores, just like we are.
Okay.
So pigs were the source of infection in Germany, at least, and Virkov did a tremendous amount of good in terms of advertising the fact that this is a disease-producing organism and that it comes from eating raw or undercooked meat. But if you cook it and treat it properly under rigorous and rigidly controlled rules for smoking, you can actually smoke the hams and salt them and kill off the parasites this way as well. Today we know we can freeze the meat and kill them this way as well.
So in 1835, what fraction of the world's population was infected with Trichinella spiralis?
Well, because it was just discovered that year, I couldn't give you the answer, but
within the next 10 years, say.
It was as high as 40% of the autopsies in New York City during the 20s and 30s. 40%. And that's mostly an immigrant population from Europe. So a lot of infection in Germany and Italy and France and Spain and United Kingdom.
So Virchow's preachings, when was that roughly?
It was in the 1860s.
So did that eventually spread around the world?
It did. And in fact, it was an anecdotal story that's worth telling now, that the animal producers of Germany took offense at what Virkov was saying, because every time he'd give a lecture, the price of pork would drop to zero. Nobody would buy any, and they were all afraid to eat it.
Sounds like swine flu in 2009.
A little bit. So they hired this veterinarian to shadow Virkov, and every time Virkov would give a lecture on this subject, this guy would get up and say, that's this nonsense. We eat this all the time. He says, this is a German national dish, raw flesh.
Sure.
We love it.
They're always skeptics.
They are. So one day, Virkov came prepared, of course, and offered the veterinarian a piece of raw, infected pork. Because the veterinarian was contending that this doesn't cause any disease. So he held up this meat to this audience, and the audience began to chant,
Eat it.
S, 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 which is the German word, of course, for eat, right? And the veterinarian backed down. He wouldn't eat it. And he was smart, because if he had, he probably would have gotten sick and he could have possibly died.
Well, maybe he already was infected, though.
Maybe, but he wouldn't have known that there's an immune system that kicks in for that. So he was definitely intimidated by Virkov. Virkov became so famous that he was elected to the German Rathaus and became a good friend of Baron von Bismarck. So they were both pathologists in their own way, as I put it. Bismarck in his, and Virkov in his.
Interesting.
Yeah, and so by the turn of the century,
By the turn of the 20th century.
Yes, by the turn of the 20th century, people were aware of the fact that if you inspected meat and found the larvae, that you could kill it by freezing.
Is that right?
Yeah.
Freeze the pork and that's it?
That's correct. Or discard that carcass, one of those two things.
So today we can just freeze our pork and we can have it red.
You can. You could have pork tartare if you'd like.
As long as it's frozen.
As long as it's frozen.
Is the pork today inspected so that it would be known? But you can't find everything.
Hence, that's a fascinating question, because that's the history of the discovery of how to inspect. You take these large glass plates and you press them together with a large piece of diaphragm and you shine a light through it, and it comes up on a screen and you can actually see the larvae in their nurse cells.
So you just take the diaphragm from the pig.
Yep.
You don't have to worry about any other tissue.
That's right.
If it's there, it's everywhere else.
That's correct.
Fascinating.
Yeah.
So do they do this at slaughterhouses? The USDA requires this? No.
Well, that's the joke, you see. That's the joke. Because the United States began an inspection system based on Virkov's...
Is that right?
Yes.
When?
Back at the turn of the century, the 1900s. Okay, let's see, 1910, 1915. But the German pork industry became so intimidated by the overproduction of pork in the United States and were afraid, of course, that they would outcompete them in Europe, that they rigged up. This is the apocryphal story of this one. Okay, the apocryphal story goes that somewhere in Germany, some meat inspector put some infected meat into the system from the United States only, of course, and that they claimed to have passed inspection, of course, in the United States. It was trichinella free, but look, here's a piece of pork from the United States, and it's loaded with trichinella. We can't trust the pork from the United States, so we will ban it.
I see.
And the United States, since that point, has had a very difficult time in exporting pork products that have been uncooked, I should say, unprocessed pork products to the rest of the world.
Is that right? Still to this day?
Yes. I would say that that's the truth.
We have a lot of pigs in the U.S. There's a huge global pig industry, as we know now. That's right. Because of this year.
Exactly right.
The flu virus originated somewhere in pigs. So,
that's right.
And in fact, we know in 1918, that's when influenza first went into pigs.
Indeed.
On farms.
That's right.
But at that point, there was no more Trichinella. Right?
That's right. So I have a story about pigs and swine flu and Trichinella at Rockefeller University with none other than Robert Shope Sr. So Robert Shope Sr. was involved in research with pig ascaris, which is a very large worm. It's about the size of the pen you're holding.
Ascaris lumbricoides?
Correct. And in this case, it's Ascaris sum, the pig ascaris.
Okay.
And apparently the egg of pig ascaris can harbor swine flu. So the egg acts as a carrier for the flu. Yeah. And that's how it goes from pig to pig. So he did a lot of work at Rockefeller with
That's the way it was thought. I'm not sure that's...
Well, that was what his findings were, you see. And then that's when he came up with his expression, the earth is covered
with pig, blah, excrement.
That's right.
Yeah, I think that it's clear nowadays that the virus is transmitted among pigs the same way
by secretions.
Secretions, sneezing, coughing, contact,
Okay.
He did publish those findings years ago. In fact, when I was in graduate school, we were taught that also, but I'm not sure how significant it is anymore.
All right, but they became known as Shope's shotes.
Shope's shotes, very good. A shote is a male pig.
That's correct.
And a female pig is a sow.
It is. Well, you're way ahead of the curve here. And the offspring are called?
Piglets.
That's right.
I like these names for male and female animals.
Indeed.
Swans and cygnets.
Yep.
Okay. Sorry, I'm going down
that's
rabbit
okay.
hole.
It's okay. It's okay.
So when did we... So I just remember when I was a kid having to worry about cooking your pork. I was only born 56 years ago. So why is this still persisting in the 50s, 60s, and 70s?
That's a very good question. In fact, it's a valid question because of unsanitary conditions on farms. Now, who wrote the best book you can think of with regards to unsanitary meat inspection and meat slaughterhouses? It was a famous
it was... now I'm forgetting the name, but I read it. Was it Orwell?
No, Sinclair Lewis.
Sinclair Lewis.
And what was the name of that?
No, I can't remember. Damn.
The Jungle.
The Jungle.
Sure.
I love that book. I read it in high school or 8th grade. Great book.
Well, things haven't changed too much, unfortunately.
It was terribly unsanitary, yeah.
Absolutely. And so there are periodic cleanups, and then there are periodic lapses back into that.
So to this day, can you find Trichinella in pork?
You can. It depends on where you look.
U.S.?
Rare. Very rare.
Japan?
Rare.
Canada?
More frequent.
Mexico?
Very frequent.
South America?
Extremely frequent.
Africa?
Frequent.
Europe?
Not so frequent, but Middle Europe, yes.
Asia?
Very frequent. China, Hunan and Yunnan provinces are just absolutely chock full.
Wow.
So if you go to the website that we're recommending all the time, trichinella.org, you can, under epidemiology, you can see maps of distribution, and you can see the Chinese map that they have. For every dot, that's 5,000 pig cases.
So in China, you need to be careful with your pork.
You need to cook it.
Wow.
You need to cook it. So, but let me take you to Rockefeller University when I was a postdoc there. And I was working with Trichinella. And across the street at Cornell University.
By the way, how do you grow it in the lab? In broth? Will it grow on a plate?
No, these will not grow outside their host.
So you have to
need
put them in mice
or rats or rabbits. Any mammal will do. Even a strict herbivore like a rabbit, you can infect them.
Can I ask you a question?
You may, of course.
Sort of not on this line, but
Please.
So where did this come from?
You mean evolutionarily?
Yeah, no, it's in nature in wild pigs?
There's a sylvatic cycle. A sylvatic cycle exists, yes.
In wild pigs or all kinds of wild animals?
All kinds of wild animals. In fact, there's a checklist of who has Trichinella that goes on for 4 pages worth of a book. I'll bring it in and I'll read it to you the next time.
So we don't know when it first got into people and how, right?
We don't,
Probably from some animal.
But let me just modify the story. And now we're up to 8 different species of Trichinella. And some of them are restricted to wildlife and some of them are crossovers into human populations. The Trichinella spiralis is the domestic strain of Trichinella we're used to. But there's a nativa, a britovi, a pseudospiralis. And then we have several other designations.
So you can find different species in various wild animals.
That's correct.
So when you go out and hunt, you have to cook your meat.
You do.
Foxes, bear, deer, they could have
very common. Not so much deer. Deer are herbivores.
Squirrels.
Herbivores.
So anything that's
a bear, definitely.
A bear.
In fact, all of the bears of Yellowstone Park that have been autopsied have trichinella.
Wow.
Every bear.
How about elephants? They're herbivores, right?
They are.
Tigers and lions?
Definitely.
Panthers, all that sort of thing.
Sure. It accumulates in the food chain. Even monkeys, though. Chimpanzees.
Yeah, they're carnivores or omnivores.
That's
whatever,
right.
Yeah.
But Jane Goodall used to say that they were just strictly omnivorous. Or herbivorous,
rather. All right.
All you had to do was biopsy the muscle tissue to find out, though.
It's right. You said that once on TWiP.
Yep.
All right. Now, I'm sorry. Rockefeller used to grow these in mice.
No, wait. There was a guy working across the street who wanted to collect the newborn larvae of this, and I'll describe the life cycle some other time, but just know that you can actually filter the blood and get these circulating stages to stick to a filter of a certain pore size. And in order to get enough blood, he wanted to do this in pigs. So he infected 3 pigs. This was at Cornell Medical School. And he sacrificed 2 of those pigs, and that was the end. He had 1 pig left. So he asked me whether I wanted it or not, and I said, sure, I'll use it for antibody production. So I bring it over to the Rockefeller animal quarters, and the animal workers there remembered Shope's pigs. Now, what do you think Shope did with his pigs when he finished with them?
He ate them.
He gave them to the workers.
And they ate them. Wow.
So they were so used to having pigs for Thanksgiving and Christmas, and he was another worker that came in with pigs. Oh, good, we get to eat again, you see. So, but of course, you shouldn't eat this pig because it's just absolutely laden with Trichinella larvae in the muscle tissue, and I couldn't possibly let them do this, right? From a public health standpoint.
Sure, even if they cooked it. Yeah.
So I kept this pig for 3 months, and periodically would take food away from the pig for a day and then skin and eviscerate about 10 mice that were laden with Trichinella in their muscle tissue, and just throw it in front of the pig, and the pig would just eat that like it was tomorrow. It was
hungry, right?
Just ate it right up. So it was definitely a meat-eating pig. And at the end of 3 months, the antibody titers were extremely high, and now I had an enormous source of antibodies that I could play with.
That's how you made antiserum.
I did. That's right. But when we sacrificed the pig, of course, all the workers were willing to help me because they thought I was going to give them the carcass afterwards.
Yeah.
And I said, I can't do it. And I said, what do you mean you can't do it? Dr. Shope did it. And I said, no, I can't do it because this is an infection. This is a worm. He says, no, Dr. Shope worked on worms. We know what they're all about. I said, this is a worm you can get by eating the meat. And they said, we'll cook it. I said, I can't possibly give you this pig. I won't be responsible for an outbreak of Trichinella. The incinerator was right there, and I took the carcass of the pig, and without their help, because they wouldn't touch me after that, I was sort of a pariah. Yeah. I put the pig down into the incinerator and disposed of the carcass, and these guys were so angry with me you couldn't believe. They were almost ready to throw me down the hole. I'm not kidding, because I have deprived them of a special treat that they had been used to getting, and I couldn't for the life of me explain to them the difference between what Dr. Shope was doing and what I was doing. But that's another story. That was just a minor blip in an otherwise very productive and happy life. So
right now, what's the incidence of Trichinella globally?
Well, it depends on where you look. If you go to China and you go to those 2 provinces, Hunan and Hainan, you have a very high prevalence.
In people, I mean. In
people, it might be upwards of 30 to 40%.
So does this kill you?
Can.
How much of the time does it kill you? You have 100 people with worms in their diaphragm. 5 people will die?
5%. Mortality rate about.
Does it get in your brain?
Well, if you have muscle in your brain, yes.
Some people do.
No, but the answer is it gets there and realizes it's in the wrong place, so then it has to go somewhere else.
So it likes muscle. But you're going to tell us the life cycle at some point, right?
Of course I will.
So it can kill you. How does it kill you when it does?
Well, part of it is migrating across the sensitive part of the brain, and the other part is migrating into heart muscle.
I see. So
I once actually gave a talk called Trichinella spiralis, an affair of the heart. Because that's what you can die from. You can die from sudden heart failure.
Right. And the other 95 people are fine. There's no apparent effect of having worms?
In fact, there might be an advantage of having worms afterwards because it heightens your immune system on both the Th1 and the Th2 responses. So there was a famous actor by the name of Yul Brynner, who you might recall.
I do.
And he acquired Trichinella by eating in a restaurant in New York. In fact, it was Trader Vic's. And he sued the restaurant and got about $120,000 as a result of his lawsuit. And he could prove that that's where they caught it because 4 people caught it, and they hadn't eaten at the same place before or afterwards for 3 weeks. And so that it had to be that restaurant. It was in the spare ribs that hadn't been cooked well. Well, the point is that he later on developed a lung tumor. And it took him 9 years to die from that lung tumor.
So you're saying his immune response was responsible for that?
That's
Maybe.
It's anecdotal. It's anecdotal and everything else, but I firmly believe that the heightened immune response to his worms had an effect on the growth of the tumor.
So do you think it's good that we don't get infected, that the 5 out of 100 preventable deaths, is that good? Or do we have other benefits that we're losing as a consequence of not?
It's, Vince, it's very difficult to say because we're not victims of natural selection any longer. So I can't honestly say whether or not we're better off or not.
But at one point, maybe humans developed with these, from the onset of the earliest ancestors, they may have had these worms and it may have helped them. It
may very well have helped them. And in fact,
fascinating questions that we can't answer.
No.
A lot of and not just worms, but viruses and bacteria.
But of course.
Some of them have been beneficial at the onset.
Well, we know a lot of bacteria that are beneficial.
Sure.
But in fact, there is a beneficial effect of acquiring one worm, and that is Trichuris. It's a whipworm. It's related to Trichinella, all right?
Trichuris. Oh, there's a very gross picture in there of Trichuris. Dick, I was looking through it today. You're very graphic.
Parasitic diseases are extremely graphic.
Trichinella, yeah.
So the point of that is that if we're free of these worm infections, then a subset of us will develop a disease called Crohn's disease. Crohn's disease is an autoimmune disease in which the immune system is directed against the epithelial cells of the large intestine.
Yeah.
But if you give those people Trichuris ovis, which is the sheep Trichuris,
right,
the Crohn's disease goes away for the length of time that the worms are present in those people.
Crohn's is an immune sort of disease, right?
It certainly is. Autoimmune.
Autoimmune, yeah.
That's right. But you have to ask yourself, what is the immune system programmed to detect and to defeat? And if you look back through our evolutionary history, of course, we have a very close association with worm infections. So the moment we got rid of all of them, we started to develop allergies to foods and all kinds of other crazy things, right? Celiac disease, you name it. And we've got all these hyper-allergic responses to things that most people living in heavily wormed areas do not get. And that's the epidemiologic basis for thinking that perhaps we're living a little bit too clean.
Now, why did you work on worms? Because you started in an era where we could control the infection, and we still can.
You're right.
By just being clean and inspecting meat. What do we learn by working on it?
I began my career studying this infection in animals that had no bacteria.
Right.
Germ-free animals.
Gnotobiotic mice.
That's right. Known life. That's what that means. And so I went to the University of Notre Dame and I got my PhD there working on germ-free mice and Trichinella infection to see if you had the host and one parasite only, you could isolate all of the effects and say whatever happens in this host is due to this organism.
Right.
Right? Made sense, right? It was simplified. Occam's razor, take the easy approach. Guess what? Turns out that Trichinella needs bacteria in order to carry out normal life inside their hosts. Some of these mice became heavily infected and others didn't get infected at all inside the isolator. But when I took the same worms out of the isolator and infected an equivalent number of conventional mice, the infection was evenly taken among all the male mice. So the conclusion that I drew, I did this 3 different times to make sure that the results were the same, similar, I should say, not the same. And each time I got erratic results in germ-free animals and very consistent results in standard animals.
Right.
So I dropped that as a project for my research because I wasn't about to go ahead and find out why this occurred. That might have taken years and years and years. So I ended up working on some other aspect of the infection, something to do with the immunity.
There are still people working on Trichinella in the laboratory?
Oh, yeah.
Many people.
Less now than there were before, I can tell you.
Are we learning how this interacts with the immune system?
Well, where I left off with it, which was in 1999, I had an NIH grant for 27 years, as you might know, and I worked in the lab on this subject. My research led me to ask questions about how the worm survives in a long-term relationship with the host. Because once the larvae are produced by the females in the gut tract, and they migrate to the muscle tissues and establish their infections there, they can stay there for as long as the host stays alive. There are no aging genes in Trichinella. Isn't that interesting?
Yeah, we mentioned that last time. And a lot of these parasites don't have aging genes.
Why should they have them? Because you have them.
Well, they just make a lot of themselves. They don't have to worry about that.
Well, they die when you die. That's the point.
Right.
But Trichinella doesn't die when you die.
Right. Crawls out, right?
It doesn't. No. It waits for a scavenger to come along and eat it.
Well, it's not waiting, but that's what evolution is.
So what I ended up looking at is the way that Trichinella maintains itself inside the modified muscle cell, which I came to call a nurse cell.
So our picture in TWiP is
a nurse cell.
It's a muscle cell. It's modified. It's called a nurse cell.
That's correct.
And inside of that single cell is a single Trichinella?
Yes.
One organism.
That's not a single cell, by the way.
It's multiple cells?
It's hard to say what it is, because
fused. It's not a normal cell, right?
It's not a normal cell, of course.
So something's but it's some kind of a cell or derived from a cell.
Re-engineered.
By the worm.
Yes, that's correct.
So it survives in there, lives in there.
That's right.
And I can't wait to hear the life cycle. Did anyone ever try or want to make a vaccine?
Oh, yeah. Trichinella? In fact, I did. And many others also made a vaccine against this infection. It's pretty easy, actually. The worm secretes things into the host during its life cycle, which have purposes for the worm, of course. Half of these are coated with a unique sugar called Tyvalose.
Tyvalose, yes. We've mentioned that on TWiP.
We have. It's shared by a Salmonella group B organisms, bacteria.
And that's protective if you make antibodies against it?
In fact, it is.
And so you could take mice and immunize them with Tyvalose and they're protected.
Or with Tyvalose-related proteins derived from the parasite.
So why don't we immunize all the pigs?
Because it's not a complete immunity.
I see.
And the argument is, of course, when you immunize, then you wouldn't be able to use immunologic detection methods to see if they're infected.
Well, we don't, do we? We
don't. Of course we don't. It's
so today in the U.S., do we look at meat? No, we don't.
We don't look at meat at that level.
What do we do to prevent worms from being in pork?
I think it's mostly an epidemiologic intervention. We do surveys on farms, and we take diaphragm tissues and serum samples.
I see.
And if they're all negative, we just declare the whole area Trichinella free. But it's a little bit fallacious to say that because the animals, the wild animals in the area have it.
And they could spread it.
And there's always a chance that it'll come into the pigs.
I've just searched Pubmed for Trichinella.
Yeah.
I've got 3,565 articles going right up to the present day.
Sure.
Look at this one. Trichinellosis outbreak in Lithuania.
Oh, yeah.
June 2009. You know what it's from? Wild boar meat.
But of course.
Sausages.
A lot in Russia, too. I actually have a lot of...
How do they know there's an outbreak? A lot of people just go to their physicians and they get sick. What kind of disease? What do you throw up? You have GI problems, mental problems?
It starts out as a diarrheal disease.
How soon after you ingest the worms do you get diarrhea?
Almost 2 days. It's not very long afterwards.
You get very bad diarrhea?
It depends on how many worms you eat, but yes.
So you go to a physician?
Or not. You just say, well, it's food poisoning, and I'll just tough it out.
It lasts a few days.
And that's it. And then nothing happens for a while.
But those worms are
for
there.
About a week. Oh, they certainly are. And then they start producing their larvae. And the larvae start migrating systemically through the blood supply and into various tissues.
If you go to your physician and he or she is smart enough to look, and as you used to do in the lab, the parasitology lab, I used to see some Trichinella worms in the stool samples.
Not in the stool samples. You won't see them.
Will this? I guess in Lithuania they found it. What can you do to these people to treat them?
Steroids.
Steroids will get rid of the worms?
It won't get rid of the worms, but it'll reduce the innocent bystander effect of the immune system, which absolutely kills you in responding to all of these allergens that the worm secretes. In fact, there's no good test to detect the actual worm itself unless you take a piece of muscle tissue and look at it under the microscope.
Okay.
Just like Owen did.
Yeah. Here's another interesting one. Journal of Immunology. Expulsion of secondary Trichinella infection in rats occurs independently of mucosal mast cell release of protease. Wow. I don't even know what they're talking about.
Well, I know the authors.
So this is a healthy field still.
It is. It is.
Not burgeoning, but still there's work going on. Here's another outbreak. Imported meat from New Zealand to Singapore. So that keeps people busy. I'm looking for basic science here.
You have to go back to 1999 for that one.
No, that's not Sylvatic Trichinella in Finland. Sylvatic meaning in the wild.
That's correct. It's probably from Nativa and Britovii. I can see the designations.
I'm looking to see what kind of animals are involved here.
Foxes and wolves and
Nativa, Britovii, Pseudospiralis.
Yeah.
Raccoon dogs and red foxes are the most important reservoir animals.
That's right.
And those can be eaten by other animals. Interesting.
Okay. Yep.
This is great. Do you want to stop there and take a little email?
Sure.
Is that all for your story for today? And then
I can easily stop here.
Would you like? It's up to you.
No, fine.
And then we'll continue another time.
I'm fine. Next time we'll cover the life cycle. How's that?
That would be great. Is that our next TWiP?
How about that?
We'll do it in sequence.
Sure.
Excellent. Well, since our first episode, we have had a few email.
How remarkable.
So let's read them. The first TWiP email ever was from Bjorn, who is in Germany. Hi, Dick and Vincent. My name is Bjorn. I am a biochemist and PhD student working on trypanosomes here in Germany.
Cool.
I'm a longtime listener of your TWiV podcast and just listened to the first TWiP show. What a great podcast. I email you to express my gratitude for your weekly commitments, and especially I wanted to praise TWiP. I think the constant stream of questions from Vincent enabled Dick to focus and bring your intended messages home.
Cool.
Yeah, I'm glad I had some use.
Oh, of course.
Thanks for your efforts, Bjorn. So that was email number 1, Trypanosomes. We'll get to those one day.
We will. In fact, we'll have some guests to come on. We have plenty of opportunities there.
Yeah, right here in New York, there are people who work on these. And
sure.
Here in our own institution,
Absolutely.
We have people as well.
You bet.
What did Josh work on? Josh Stillman, who was on TWiV last time.
He worked on neurocysticercosis.
Yeah, we'll have him on, too. That'd be great.
Yep.
Etienne wrote, great first episode, but once a month doesn't seem enough. Maybe every 2 weeks.
Thanks, Etienne, but, maybe after I'm finished my teaching obligation, we can do it.
Well, I told Etienne that it depends on how popular it is. If no one likes it, we're not going to do it more than once a month. So we'll see. Tell your friends, right?
Absolutely.
Hussein wrote, a very promising first episode. Thanks very much. Looking forward for more episode and hope it becomes more frequent.
Oof.
Another one.
Oof.
Okay, Mark Pelletier of Futures in Biotech, a TWiT podcast. Great job on TWiP. Despommier's story was great. Your audio was excellent, too. There should be a History of Science podcast out there. Would be a lot of fun. It would be. I bet you would be good at that. Do you know more history besides microbes?
I do, but we know someone here who actually has had a rich history of that.
Rich Kesson.
Rich Kesson. He could come on here and regale us with lots of stories.
Well, we're going to have him back on TWiV.
Yeah.
Maybe we should have him on TWiP. Does he know parasitology history? But no one knows it better than you do. So
that's not true, but...
Sophie writes, hey, Dick and Vincent. First of all, I want to say that you guys do a great job, and I got very excited when I heard you also wanted to make a podcast about parasites. You guys are great, and I learned so much from both podcasts. I'm studying biotechnology third semester at the Technical University of Denmark, but I'm planning to go into immunological bioinformatics so I can use it for my studies.
Cool.
I have a question about the size of parasite genomes.
Oh, okay.
I'm reading the newest edition of Brock, which you guys mentioned in the last TWiP.
Right.
That's TWiP 1. And I'm afraid I got a bit confused. On page 354, it states that, apart from the strange case of Trichomonas, parasitic eukaryotic microorganisms have genomes containing 10 to 30 megabase pairs. So when you say that they have larger genomes, is it only compared to related organisms, or do they have bigger genomes than expected for their complexity? Maybe it is just Trichomonas that has a very big genome, but I would really like you to talk a bit more about it. Thank you so much.
Sure. Well, I can answer that right now, at least with one example, and that is that Caenorhabditis elegans, which is a nematode, but a free-living nematode, has 19,230 genes as far as I remember.
Okay.
And if you compare that to Trichinella...
So here I've got up a sheet with Trichinella. Where is it? Where's Trichomonas? Here, 70 to 90 megabases. But that's not as... There are others that are as big. Look at Schistosoma is 270 megabases.
Exactly.
Huge. Onchocerca, 150.
Yep.
So the Trichomonas, and then what else do we have? Trypanosomes are 30 to 40.
Right.
Toxoplasma 65. Eimeria 60.
Yeah.
Some of them are smaller. Tyleria is 8.
So who has the largest genome of any organism on Earth? Orchids.
Yeah. What's the size of that?
Huge.
Bigger than human
they're
genome.
Enormous. They're just enormous.
It takes a lot of genes to be beautiful.
I guess.
So did we say on the last TWiP that parasite genomes were generally small?
No, I think what we tried to say was that if you had to guess, compared to the free-living equivalent, whether a parasite had a larger or smaller genome than its free-living counterpart, what would you have guessed? And the answer is they're bigger. Right. They're not smaller.
Yeah, compared. That was a comparative analysis.
That's right. So Trichinella has a bigger genome than Caenorhabditis elegans.
Yeah, these are quite big. They're all bigger than viruses, except for the Mimi viruses, which are 1.2 megabases.
Right.
But there's a lot of genome sequencing. Although I don't see T. spiralis here. Is it sequenced?
Not altogether.
Pieces.
Yep.
Because this paper I have is from 2005. It's called Parasite Genomes. It's a summary of all the genome sequences.
Ross Koppel, I know Ross pretty well.
Yeah. Okay, that was Sophie. And that's it for our email today.
Okay.
And your pick of the week was...
A movie
called Devil and the Doctors.
Excellent.
I don't have a pick this week. I'm sorry.
That's a good, by the way.
Devil and the Doctors.
Oh, yeah.
All right. Let me give you some information on TWiP. First of all, if you want to send us questions, send them to twip@twiv.tv. It's a little hard. Or our website is microbeworld.org/twip, and there's also a place there where you can add comments.
Mm-hmm.
So either way is fine. Send us your questions.
Yep.
And we're happy, or Dick is happy to answer them.
Try to answer.
I just listen along.
I'll give it a whirl.
We are now on iTunes. Did you know that, Dick?
Very cool.
I submitted the podcast, and it's accepted at iTunes. So now we have TWiP and TWiV. So you can go there, and there's a link at microbeworld.org/TWiP to subscribe in iTunes. And it's great if you can subscribe. It helps us, and then you get automatically the episodes as they're posted. And if you'd like, post a comment. I think there's one comment in TWiP on iTunes. It says, "My second favorite podcast after TWiV." That's great. So go there and leave us some comments. And that pushes us up on the Apple standings so we get on the front page. That would be great. If you like TWiP, tell others about it. Spread the news, because if we get a lot of people listening, we'll do more.
Absolutely.
If nobody listens, we'll do less.
Well, I don't know. I'd still do more.
I do one a month because I enjoy listening to Dick talk, and I'm learning.
Yep. That's the deal.
So TWiP is hosted at microbeworld.org/TWiP, but we're also members of sciencepodcasters.org and promednetwork.com, as well as TWiV, which are websites where you can find other high-quality science podcasts from a wide variety of fields. That was great, Dick.
Hey, Vince.
Thanks for coming over.
My pleasure.
Trichinella.org.
Right.
That's the one for this series.
That is the one.
And you can find me at microbeworld.org/TWiP, or if you want to follow me on Twitter, although I don't talk about parasites on Twitter, at PROFVRR. Thanks, everybody, for listening. And you have been listening to This Week in Parasitism, which is all about eukaryotic parasites.
Indeed.
Thanks for listening, everyone. We'll be back next month. Another TWiP is parasitic.